this GE technology, that is DNA, gene editing technologies developed in different across the world in the following crops. And this is the horticulture crops. You see in Andhra Pradesh, 18 lakh hectares and horticulture crops and 400 lakh metric tons of horticulture produce we are producing. And the government of Andhra Pradesh is taking many steps to make Rayalaseema as a horticulture hub by investing 1 lakh crores of rupees and 60,000 crores with the private investment and 40,000 crores from central and state governments. And this is the need of the work because the gene editing and at a molecular level. So this important horticulture crops which I mentioned. So definitely this technology will help and we will have a collaborative research projects in future and exchanging of our faculty, students, and availing the facilities at our center by signing MOU. Definitely and because this is the gene technology, growth and architecture, flowering and reproduction. So for the growth and architecture, for high density planting, we can have canopy management, canopy branching, canopy, suitable high density planting, flowering and reproduction, alternative flowering time, floral lifespan and flower shape, and fruit and vegetables, especially fruit color, ripening time can be reduced, calmness, flavor, metabolism, and seed size. All whatever we want, we can incorporate in DNA and we can regenerate. And shelf life, we can increase the shelf life by DNA, gene editing, and biotic stress, abiotic and biotic stress can be induced. So whatever the quality we require within a short time, we can create in DNA. And you see many things are there, many technologies also there, that is ZFNS, talents, CRISPR, Cas, base editing and primate editing tools are there, and many advantages are there with the DNA editing. And CRISPR is, I think you are very strong and very expert in CRISPR technology, that gene editing in tomato, potato, I think many vegetable, brassica, cabbage, and cauliflower crops. You see, really, our students and our faculty should update our knowledge, and in future also we have to work for the gene editing. That's why, and by integrating artificial intelligence with genome editing, PAM genomics, transcriptomics, metabolomics, single cell RNA sequencing, high throughout phenotyping. So Dr. Janga's research in pan genomics and single cell RNA sequencing provides valuable opportunities for collaborative capacity building. Precision editing opens doors to rapidly domestic resilience, underutilized wild species into viable commercial crops through de novo domestication. In this context, I would like to give clear directions to our scientists to build advanced transformation and editing infrastructure, focusing on priority crops, precision breeding, and AI integrated genomics. The future of horticulture is not simply about growing more plants, but engineering better plants for a changing world. With this vision, I once again extend my warm welcome to Dr. Madhusudan Reddy Garu, Janga, and express my sincere appreciation to Texas Tech University for this valuable academic collaboration. I am confident that today's Global Connect series will open new avenues and will contribute to the advancement of the horticulture in Andhra Pradesh, India, and globally. Thank you, sir. Thank you. And welcome to Dr. YSR Horticulture University. Your expertise and our knowledge will be definitely utilized by our scientists and faculty and students of Dr. YSR Horticulture University. This is the throughout the state, almost 41 institutes from different institutes, different scientists are connected, and you can have thorough sharing your knowledge, and any queries can be they will ask, and you kindly share your knowledge. Thank you once again to Dr. YSR Horticulture University. Kindly proceed.
Thank you very much, sir, for your motivational words and vision for global collaborations. Now I request our guest speaker of today's program, Dr. Madhusudan Reddy Garu, to deliver his talk on plant transformation and genomic editing technologies and their applications in horticultural crops. Sir, please.
Thank you, Vice Chancellor, sir, for the great introduction on the GINA meeting, and Padmalata Madam for arranging this seminar. So I'm going to share my screen. I hope you can see my screen now.
Yes, sir. It is visible, sir.
So today I'm going to talk about the plant transformation and the genome editing technologies and their applications in crop improvement. So this is actually a vast topic. It's not easy to cover in one lecture. So I try to quickly cover these several important aspects, and utilizing this knowledge, you can think about your specialized crop and how you can bring these aspects into your own crop. You can think in that way, and in future you can apply these technologies to your interested crop. So why we need to improve these crop plants, especially if you see the horticultural crops, we are having a lot of limitations in terms of a longer shelf life. On top of that, we have growing demand. Population is increasing, and at one side we have less land, and the water supply is limited right now. So because of that, we have growing demand, and the greater demand for nutrition and food, and need to produce more from limited land and water. We need to adopt this one. And we also have this consumer demand, like better nutrition, improved taste and quality, and longer shelf life. And on top of that, we have a lot of environmental challenges right now: heat and drought, salinity, and unpredictable weather. One year we have drought, other year we have floods, and on top of that, we have these emerging pests and diseases. So I just saw this one in the news recently, like this is published just 2 days ago. So this picture is taken 1 month ago on the Godavari River, which should not be drying like this. This is from Maharashtra, and this is from the AP water resources. We can see the major and medium and overall total. So last year we have 76% of the reservoirs are filled. This year we have only 54%. So this year we have severe drought. So all these challenges are affecting us in the crop productivity, and even for the horticultural crops, it is affecting to improve the yields. So breeders and the researchers always trying to improve the crop plants and develop the suitable varieties by doing the genetic modifications. So genetic modifications is not new. It has been used in varying farms for centuries. So initially we had
ఏమన్నా
these
లూజ్ అయిందేమో చూడు అతనికి అస్తమానం కదులుతూ ఉంటే లూజ్ అవుతుంది అది వెనక.
Please.
yeah. So for example, we have this making watermelons that are sweeter and have fewer seeds. So for this, we can apply any of these technologies. So if we have a particular wild type varieties, we can easily cross them through the selective breeding. If we don't have the particular genes or particular trait in even in the wild type, we cannot bring those in or introgress into your interested line. So for that, we have to do the mutation breeding. So mutation breeding, they started applying from 1920s. So this is random, unpredictable, but relatively fast compared to these other technologies. Similarly, this transgenic breeding came in 1980s. So interesting thing about this transgenic breeding, this is completely different from the selective breeding or the mutation breeding. In these two process, we are utilizing only the limited gene pool. Whatever is available within that species, we can apply this technology. But whereas for transgenic breeding or transgenic, to develop the transgenic plants, we can bring any of the genes. For example, we have the BT cotton. In the BT cotton, we are getting the genes from the bacteria and inserting those genes in the cotton. So through breeding, that kind of feasibility we do not have. So we have to apply this transgenic technology wherever it is needed. Now, after that, we have the genome editing. And with the genome editing, right now we have greater capacity. We are applying the next generation sequencing technologies. Almost all the crop plants and even the many of the horticultural crops, right now we have the full genome sequences available. That means we know how many chromosomes we have, how many genes we have on each of those chromosomes, and for many of those genes, we have some information from either model crops or from the other crops. We can get that information and try to improve the crops by editing those genes. So why genome editing? So genome editing makes these targeted modifications in any of the genes associated with the important traits. For example, in the previous slide, I mentioned about the watermelon. If you want to increase the sweetness or develop the seedless watermelon, you can if you know the genes, you can simply knock out those genes and you can develop the trait easily in your interested genotype. And it is not replacement for the breeding, it only complements conventional breeding. So that is the important aspect. It speeds up the process. Overall time, we can reduce applying this genome editing technology. So what are all the different traits that we can apply? As just now Dr. Vice Chancellor Sir said, many of the different traits we can improve. Some of the traits I have given here. So for horticultural crops, mainly we have these consumer traits like improving the nutrition, flavor, texture, shelf-life, reduced browning. These are the important traits that we can improve. And from the farmer side are to improve the crop yield, disease resistance, insect resistance, plant architecture. These are the traits we can improve. And for the climate and sustainability side, drought, heat, salinity, water use efficiency, and reduced pesticide usage. These are the different traits we can apply, and there are many more other traits also we can improve applying this technology. So this is from one of the Nature Review article. So here they are showing that four different physiological factors that count for the produce quality, like flavor, texture, shelf life, aroma. If you see here, normally farmers harvest during this unripe stage and during the transportation it ripes. But especially like tomato, we have a lot of information available. We know what are the genes that are involved in the ripening. If we can slow down the process of ripening, so this post-harvest waste will be reduced. So with that introduction, I would like to take you to today's present topic. So I would like to give some overview of what I'm going to talk. So I'm going to talk about the plant tissue culture. So plant tissue culture, plant transformation is completely different. Plant tissue culture is a process where we can generate the plants from any of the tissues of the particular species. For example, we have banana or any other crop if we take. So complete regenerated plants we can develop, and many like millions of plants we can generate within with very limited time and with very limited resources. We can develop those plants. So DNA delivery and plant tissue culture together, if we incorporate, we can generate this transgenic plant. What it means is, so normally the plant cell will have certain DNA and we are inserting the foreign DNA. So we need to develop that DNA delivery method. Once we have the DNA delivery method, we can apply these two technologies together, plant tissue culture and the DNA delivery method together to develop the transgenic plants. And I'm going to give some examples of plant transformation methods because each of the species might require different plant transformation method. So using those examples, you can understand and learn that what are the different technologies available and how we can adapt those particular methods into your own interested species. So after that, I'm going to talk in the second part about this genome editing in cotton and potato. And I would like to show one example starting from the gene discovery to genome editing in cotton. So first topic is the plant transformation. So for plant transformation, we need these two aspects, plant tissue culture and DNA delivery. If we combine those two, together, we can generate the transgenic plants. So what is plant tissue culture? It is a technique to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture media. So this is all happening in vitro. So as you can see, this is the laminar fluid hull. So where it maintains the sterile conditions, in this laminar flow hood, we can subculture these tissues here. So these are the Petri dishes, they contain the media, and we can place our tissues there. And from those tiny tissues, we can regenerate many plants. So this is the growth chamber you can see here. This growth chamber is required to maintain the optimal conditions to grow the plant tissues here. And culture media also important here, which contains the nutrients, hormones, and water that are necessary for the plant tissues to grow. So generally plants have this totipotency, so that means we can regenerate the whole plants by taking any of the plant tissue. But the important thing for the plant transformation is we need to identify the right tissue from which we can generate the transgenic plants quickly. We don't want to spend 2 years or 3 years in this process. As soon as possible, we have to be able to generate the transgenic plants. So we can use, depending on the species, we can use, in some species, we can use the seed, mature embryo, immature embryo, callus, hypocotyls, cotyledons, petioles, cartilage nodes. These are the different types of tissues we can use. If we are bringing the tissues from outside, they will have the pathogens. So we have to surface sterilize them. So for example, I'm doing transformation on the rice, or if I'm doing transformation on tomato, I need the seeds. So seeds are coming from the field or greenhouse, so I have to sterilize those seeds. So we have these disinfectants. We can apply sodium hypochlorite, ethyl alcohol, or chlorine gas. These are the popular methods, and you need to optimize the concentration and how long you treat those tissues. Depending on that viability of the seeds, you have to test and identify the optimal conditions for that. So once you have that, you need to identify the growth media. Medias can be either liquid or semi-solid. Nowadays, most of the people are using the semi-solid media to reduce the contaminations and easy of the growing the culture. So that media contains the inorganic salts, vitamins, carbon sources. And we need to have the growth regulators. These are the growth regulators. They help for the either to go into the, for example, you need to produce the somatic embryos. We need to use the auxin such as 2,4-D. And if you want to produce the organs such as shoots, you need to use the cytokinins. Or if you want to produce the roots, you have to use the NAA or other auxins. Similarly, sterile conditions you have to maintain, and frequently you have to subculture, either to move the cultures into the next stage or changing the developmental stage from one stage to other stage. We need to develop applying these different hormones. So here on this slide, we can see the types of in vitro plant regeneration. Figure A shows this mode of axillary branching. So using this method, we can make millions of plants within months. And figure B, you can see advantages, roots are forming, or advantages, shoots are forming. So generally we don't see on the leaf, you cannot produce the shoot, or from the leaf, you cannot produce the plants won't produce the roots. But in the tissue culture process, by applying the hormones, you can change from one stage to another stage. You can easily convert these tissues. And in figure C, you can see de novo origin of regeneration from the unorganized callus. So from the single somatic cell, true callus is formed. If you apply the cytokinin, shoots will come. If you apply the auxin such as NAA, we can produce the roots. If you apply the 2,4-D, we can produce the somatic embryos. And we have this another method, advantages origin of mode of somatic embryogenesis. So this is, imagine like leaf tissue. From the leaf tissue, directly these somatic embryos are forming here. So these are the different types of plant regeneration methods. So plant tissue culture is very old method. As you can see, this work is done in 1958 itself. So in 1958, for the first time, in vitro culture of the carrots they have performed, and they have taken these carrot tissues, made the cell suspension. When they applied the 2,4-D, these cells clumped together, and they have generated this hard stage embryo. After removing the 2,4-D, these embryos got matured, and they could regenerate the whole plants from these tissues. So 1958 itself, they started working on these plant tissue culture methods, and they learned how to develop these plants at that time. Right now, the basic tissue culture is applied for many of the purposes, mainly for the propagation of the plants, like banana tissue culture. All of you might have heard this. There are many companies in India, and even some of the universities are developing these banana plants and giving that to the farmers. So within months, you can generate millions of banana plants using this plant tissue culture. So if you are interested in your species, first you need to fix the plant tissue culture process, where you can take any of the tissue and regenerate the plants into fully developed plants. Once you have that protocol, then you have to work with the DNA delivery to generate the transgenic plants. So there are majorly two different types of methods available. We divide them into two categories. Non-biological based transformation, which is the direct method we can call. So no biological organism is involved in these methods, but they are not working that well. Only the particle bombardment will work to some extent. But majority of the choice for majority of the researchers is the biological gene transfer, which is utilizing the agrobacteria mediated transformation. Or there is a recent article published in 2022. You can even use this other bacterium as well to transfer the tDNA. And these are the comparisons. Like if you see the agrobacterium mediated versus particle bombardment, which is also called the biologic or gene gun. So this is host dependent. What it means is it has to interact. So bacteria and your plant species has to interact. If they have the well interaction, tDNA transfer will be easy. And here it doesn't depend. It is a non-biological method, so there is no interaction here. And vector requirements, these are all the different types of requirements we need to have and comparison. But majority of the choice right now is the agrobacterium mediated method. So this picture here, it shows the particle bombardment, which we call the gene gun. So this is the actual instrument here. So these two tanks are filled with the helium gas, and applying the helium gas and these pressures, we can deliver the tiny DNA into the plant cells. So plant cells will be placed here on this shelf, and we place the DNA into these carriers, and with high pressure, it will send the DNA into the plant cells. But majority of the choice for all the researchers is the agrobacterium mediated plant transformation. So I'm going to explain more details about this agrobacteria mediated plant transformation. So on this picture, you can see here, these are the cells of the bacteria. So this is the bacteria, and here I have given the representative plant cell. So plant cell has the cell wall, and inside the cell, we can see the nucleus. Within the nucleus, we may have, depending on the species, a lot of chromosomes. So here, one of the chromosome you can see, this red one is the tDNA. So tDNA means transferred DNA. So tDNA, which is in the red color. So randomly got inserted into this DNA. Normally, this plant doesn't have the DNA, but this agrobacteria is able to connect with this and send that little bit portion of the DNA, and that entered into the nucleus. After entering into the nucleus, it got incorporated into one of the chromosome. So that's how this agrobacteria mediated plant transformation will work. So here you can see in the tissue culture plate, the cotton plants are getting regenerated. So for plant transformation, we need two different techniques. One is the plant tissue culture. When you have the capacity to regenerate the plants, at some of the stages, you need to be able to insert the DNA, which is delivery of DNA. So insert DNA into the plant genome. And this inserted DNA is called the transgene, and resulting plant is called the transgenic plants. And many of you might have heard GMOs, which is the genetically modified organism. At this stage, it is not a normal plant cell. We inserted a foreign DNA. That's why we are calling that as genetically modified. These are the chromosomes. These are the genetics here. We are changing this. Something we are changing here. We are foreign DNA, we are inserting. That's why we call this as the genetically modified organisms. Transgenic plants. So you can see here, transgenic plants are developed for both research purpose and for crop improvement, either to test your hypothesis, or if you already know the genes, you can directly apply this technology and improve the traits such as abiotic stress, biotic stress. For example, BT cotton developed through this process, yield improvement and quality improvement you can apply. So what is Agrobacterium? Agrobacterium, as I mentioned before, it is a bacteria which is gram-negative soil bacteria. It is actually a pathogenic bacteria. It can cause either crown gall disease or hairy root depending on the species. Agrobacteria tumefaciens can cause the tumor. It contains this tumor-inducing plasmid, so it causes these tumors. As you can see in this picture, this is how it makes a lot of tumors on the plants. And Agrobacteria rhizogenus is the roots producing bacteria. So in these images, you can see the overall picture of this crown gall disease. So crown gall disease has been found to occur approximately 40 economically important crops. Infections may occur from both the soil or from the infected propagating material, such as grapes. In grapes, they try to prune every season. So during that pruning process, if they have infection to one plant, they are carrying and they are spreading to whole field. So agrobacteria, you can see here, bacterial genome will be there. On top of that, we have this TI plasmid. On the TI plasmid, we have these borders called left border, right border. Within these borders, we have these three different gene cassettes, auxin, cytokinin, opine. So this LB to RB is the important piece, which is the tDNA, transfer DNA. This one is getting transferred into the host cell upon finding the plant. So this bacteria can interact with the plant cells and transfer that tDNA into the one of the chromosome. After that, because of these genes, the cells lost their capacity to withhold. For example, it infects near the stem. The cell will not maintain its status. Rather, it is continuously dividing, which is like the cancer, continuously dividing and makes this crown gall. So that's how they identified initially, and later on they studied what is the mechanism, how it is able to transfer this tDNA process. So here in this picture, we can see this is the plant cell here, and we have the bacteria, Agrobacterium tumefaciens here. There are several steps. Initially, bacteria will come closer and identify the plant cell. Upon identifying, it makes this attachment. Once it attaches, the signaling molecules will come. Phenolics or sugars will be there. Due to that signaling, it will receive, and then virgin induction will occur. Upon virgin induction, it will process this tDNA. From the left border to right border, it will process. After processing, this tDNA will get transferred into the plant cell. Once it enters into the plant cell, the tDNA will target the nucleus. So it enters into the nucleus and randomly somewhere it will get integrated. This whole process they identified. After identifying this process, we are utilizing this process to make changes and deliver our DNA into the plant cells. So plant transformation depends on successful plant transformation relies on the ability to introduce the DNA into the single cells and then generate whole fertile plants from those transformed cells. So the first step here, we are utilizing the agrobacteria for the DNA delivery. So as we have seen in one of the previous image, we have seen the TI plasmid. So this TI plasmid will contain the left border to right border. Within this border, we have the oncogenic genes, auxin, cytokinin, opine. So now researchers have removed these 3 genes and incorporated our choice of genes. So for example, if I'm making the BT cotton plant or BT brinjal, I can replace one of all these genes and incorporate one of the selectable marker and insert that gene cassette, Cry1AC or Cry2AB, such genes we can incorporate between the left border and right border. Then agrobacteria will process that and integrate into the plant cell. So upon DNA delivery into the plant cell, once it get integrated, we have within a tissue, we have thousands of cells, millions of cells. But how are we going to select our cell? Not all the cells are going to take up that tDNA. Only very few cells are going to take up, but we need to select. So to select those cells, we need to apply the selectable markers. For that, we can use either antibiotics, herbicides, positive selection, or even in some cases, people use the reporter genes such as GUS, all these different markers, GFP, green fluorescent protein, or the red fluorescent protein. So I'll give one example of this antibiotic. For example, kanamycin is an antibiotic. Normally, it is binding to the 30S ribosomal subunit to inhibit the translation, which means protein synthesis will be stopped in presence of the kanamycin in the tissue culture media. But if the cell has this NPT2 gene, this is going to generate the protein, and that protein is going to interact with the kanamycin, and this kanamycin effect will not be there in that cell, and the cell will behave normally and it will continue the translation. So protein synthesis will occur normally. So as long as we have this gene in presence of kanamycin, protein synthesis will occur. If we don't have this NPT2 gene, protein synthesis will be stopped. Eventually, cells will die. That's how we can select these plant cells. And they have, each of them will have different type of mechanism here. So you need to identify the suitable selectable marker for your interested plant species. So once we have the DNA delivery, and then we have the selectable marker established, then you need to regenerate the plants from those single cells. So how do we regenerate? We have basically 2 different methods. Either we can go through this somatic embryogenesis, or we have another method called organogenesis. So these both, and again within this there are 2 generations. So the older generation, which is the first generation, where people used to apply hormones externally and generate the plants. Now we entered into this second generation technology, where we are utilizing the morphogenic genes to improve the recovery and regeneration of the transgenic plants. So older technology is applying the hormones externally. Now within the tDNA, we can incorporate the hormone-related genes. Those are the developmental regulatory genes we call. For example, some of you might have heard this baby boom whistle. These 2 genes, if they applied in the maize plant, so normally the maize transformation is relatively easy. You can get 5 to 10% efficiency if any 1 or 2 year experience person, if they do, they can easily get 5 to 10% efficiency. That means if they utilize the 100 embryos, they can generate 5 to 10 plants. In this case, when they use these genes, they used only 45 embryos, but they could generate 380 independent transgenic plants. So the efficiency increased so much, not only the efficiency, but it is normally it takes with the older method 6 to 8 months. This method is taking only 1 month to generate the transgenic plant. So that is baby boom whistle. Along with that, we also have this GIF, GRF technology. So this they applied on several different crop species as well. It's not that efficient as the baby boom whistle, but even these DR genes also we can apply and try within your interested genes. So at our plant transformation facility, I worked at the University of Missouri, and right now at Texas Tech University. At both places, I have the capacity to produce various transgenic plants, or even to develop the new transformation protocols we can also establish for any species. But these are the established crops: maize, soybean, rice, sorghum, brachypodia, switchgrass, tomato, cotton, for these crops, we can right away provide the transmission services if anyone is interested. So I would like to take you through all these different crop species and what are the variations among these. Why one single technology will not work for all these different crops. We will understand that after looking into this data now. So here on this slide, we can see the cotton transformation. It takes 9 months to 13 months to regenerate the plants. This is all with the first generation technology. We are still working on with to apply the second generation technology where we can faster this time period. So here we generate the seedlings, 10 to 12 day seedlings we take, cut these seedlings, all the hypocotyls, petioles, cotyledons we can take, infect with agrobacteria, and that will go through callus induction media. Then somatic embryogenesis, you can see this is the tiny somatic embryo and it is getting matured and getting into whole plant, and we regenerated the whole plant. Let's go to the sorghum transformation. So here you can see this is the panicle, 10 to 12 day embryos we take. After sterilizing the seeds, we isolate the immature embryos. 10 to 12 days after pollination, we isolate these embryos and treat with agrobacteria. Agrobacteria will transfer the DNA into the cells, some of the cells. After 3 days, we move them to the resting, which contains the antibiotics to kill the agrobacteria. After that, it will move into the selection. For this, we apply herbicide or the hygromycin selection also will work best for sorghum. So after selecting the cells that have taken up the tDNA, from those cells, we are regenerating the plantlets. So this takes 6 to 7 months. So there is difference between the cotton and sorghum. Now soybean transformation, if you see, this is the mature seeds. We germinate for 1 day, split them into half, and then from these cartilage tissues, you can see how we are regenerating the tissues, the whole plants. So this will take 4 to 7 months. So maize transformation is equal to the sorghum. Immature embryos we collect and treat them, generate the transgenic plants. Time taking for this is 6 to 7 months. Now rice transformation, this is again different. So initially we induce the callus, so scutellum derived calli we develop from these mature seeds. Once we have the callus, we are doing the co-cultivation with the callus. So agrobacteria and this callus we are mixing together. After 3 to 4 days, we move them into the resting, eventually to selection. Only the transformed cells are growing here. Once we have enough tissue, we send it to the regeneration and then develop the whole plants. So like that, we have established these protocols, or we adopted from other researchers and developed further all these protocols. If you are interested generating transgenic plant, first you need to establish the plant tissue culture method for that species, identify the DNA delivery method, then establish the selectable markers. Eventually, you have to regenerate whole plants from that. So that is the first part. Now I'm going to enter into the second part, where I'll show the gene discovery to all the way to genome editing in cotton. So for this, I take the examples that genes regulating gland development in the cotton. So here, first I'll show how we identify the genes. So for this, we applied the comparative transcriptome analysis for identification of genes regulating gland formation. Once we identify those genes, we established the CRISPR-Cas9 system in cotton and applied the CRISPR-Cas9 system to characterize those genes. So as all of us know, cotton is mainly grown for the fiber. Annually, it is estimated cotton fiber production is 25 million metric tons. Cotton seed is the byproduct. As you can see, this is the cotton seed file from the mill outside of the Lubbock. They captured this picture a decade ago. So we produce more seed compared to the cotton globally. 47 million metric tons, which contains a lot of protein. If we can utilize this protein, we can feed 575 million people per year, which is approximately 50 grams per day. But cotton seed is not getting utilized very well. It has this gossip ball, which is toxic to the non-ruminant animals. including pigs and chicken, humans, fish, all the non-ruminant animals cannot digest this cotton seed. So only ruminant animals can utilize this, but there also some issues are there. It's binding to the important amino acid like lysine and reducing the meal value also. So why do these cotton plants have these glands actually? And why do they have this toxic terpenoid compound called gossypol? So that is to protect the plants against various insects. So these are the different leaf and other cotton plant parts. Even in the bowl, you can see a lot of tiny dots are there. These are the dots, they are the glands. They are filled with the different terpenoids. So naturally, there are mutant varieties. So those mutant varieties do not have any glands. But if farmers grow that in the field conditions, they are not going to get any yield. So all the traditional and non-traditional insects are also attacking the cotton and destroying the crops. So that's why farmers are not growing that anymore. So first we wanted to identify what are the genes that are regulating these gland formation. Since we have these glanded variety and glandless near isogenic line, we want to compare, we want to perform RNA-seq on them and identify the genes involved in the gland formation. So in this picture, you can see that 14 days post-anthesis and 16 days post-anthesis embryos, we isolated and captured these images. We have two different genotypes. One is the STVGL, capital GL is the glanded. So at 14 days, we don't see any glands here, but by 16 day, we can see these tiny dots here. These are the glands. So glanded variety is able to produce by 16th day, we can see the glands. Glandless variety is not able to produce glands at any stage. If we see, it's completely glandless throughout the plant. So 14-day embryo and 16-day embryo, you don't see any glands here. So for the RNA-seq analysis, we took 6 total tissues. Glanded, glandless, 2 different conditions and 3 different ages, 14 day, 16 day, 32 day old tissues we took and performed the RNA-seq analysis, identified these differentially expressed genes. So these are the genes that are highly overexpressed in the glanded, and these are the genes that are expressing only in the glandless. By the age of 16 day, more genes are differentially expressed. By the 32 day, and many more genes are differentially expressed. That's because a lot of other developmental activities are going and we can see these changes. But we are more interested on these 14-day. Here you can see the heat map of the gene expression, and this is the volcano plot. What is the difference? And these are some of the important genes that we labeled on this image. So when I looked into those 33 genes, we found interestingly many of the transcription factors. So these are the important transcription factors we found. So genes encoding these putative transcription factors were tested for their role in the gland formation using there is a quick dirty technique in the cotton. We can apply this virus-induced gene silencing. What it means is we can silence these genes and see. It is not a stable phenotype. We can see to some extent, we can see the phenotype. So we applied this technology and tested. So this is, for example, CLAV1 gene is the chloroplast. This is synthesizing chloroplast biosynthesis. It's helpful for biosynthesis of chloroplast. When you silence this gene, you can see albino leaves are emerging here. So we infiltrate the agrobacteria here, and that will transfer the virus, and eventually you get these white ones. And this is the empty vector, which means we are not silencing anything here. So interestingly, out of the 14 tested, with the 3 genes, we could find significant differences. Reduced amount of glands or completely glands are absent with the CZF3. Compared to this empty vector control, you can see a lot of tiny dots. I have more pictures here under the microscope. So CZF1 gene, CZF2 gene, CZF3 gene silencing, once you can see under the empty vector, we have more glands here. So we performed that. Also, we could identify the phenotype. Also, we performed the HPLC analysis. there are several different types of terpenoids available in those glands. Gossypol, hemigossypolone, heliocytes 1, 2, 3, 4. These are the terpenoids. And you can see that this one is the empty vector. CGF1 gene silenced leaves. CGF2 gene we named, CGF3 gene. All these significantly reduced compared to the empty vector. So after identifying those 3 genes, next we wanted to see what is the cause actually. Why these glandless mutants, what are the changes at genetic level? So we compared the glanded versus glandless sequences. So glanded sequence, if you see, this is the promoter region, CDS coding region, and that's terminator for the CGF3. And this is since cotton is a tetraploid, we have A genome and D genome. For the same gene, we have these copies here. So for the ACGF3, we have this transposon, 5.1 kb transposon got inserted in the coding region. Because of that, this gene got disturbed, and we have the mutant phenotype here, glandless phenotype. At the same time, we have this D genome. In the D genome, glanded, we don't have any mutations, but in the glandless, we have several mutations in the promoter region, and in the coding region also we found a couple of mutations. Due to these changes, we expect that glandless phenotypes are evolved. So conclusion from that RNA-seq is we performed RNA-seq on the 6 different tissues, 14, 16, 32-day post-anthesis, glanded versus glandless we compared, identified 33 genes that are upregulated in the 14-day glanded. And out of them, we found the transcription factors, performed Wix analysis, and found the 3 genes that are regulating the gland formation. So next, we wanted to apply this genome editing. So genome editing, CRISPR-Cas9 system is much easier compared to the TALEN, zinc finger, or other older technologies. So all it needs is this Cas9 protein and this sgRNA. So as long as you have these 2 reagents, one is the protein, other one is this single guide RNA. If you have these, initially the Cas9 and this RNA complex will form, and it will search entire genome. It will enter into the nucleus and search entire genome and identify the target sequence like this. It will match to the 20 base target sequence. Whenever it finds the target sequence, it has 2 domains in the protein. They will make the double-stranded breaks here. Whenever double-stranded break occurs in the chromosome, in the DNA, cells has to repair it. And for repair, there are 2 mechanisms. One is the non-homologous end joining, and other one is the HR, homology directed repair. This is the majority of the times this non-homologous end joining will occur. Due to this, few deletions or insertions will occur. And homologous recombination occurs only at particular cell division stages it occurs. Either it repairs from the donor template or from the sister chromatid. It can do that repair. So to understand that whether all this technology, they started in 2010, and by 2015, they successfully developed in the monocots and even in some of the important model crops, they could establish the system. At that time, still the cotton is new to this, and we wanted to see how it works in cotton. So we have in the lab, we have this stably expressed GFP line. So what it means is it is a transgenic plant. We inserted this cassette, promoter, GFP, and terminator we have. So this is a green fluorescent protein. So this cotton tissue is expressing in the green color. So after genome editing, this picture you can see half of the tissue, the gene is not expressing. Only in the half of the tissue it is expressing. This, fortunately, we found this chimeric tissue here. So overall, we found these 3 different target sequences. So these are the target sequences, 20 bases, and we have this PAM sequence NGG for the regular Cas9. So 3 different targets I tried. So right now we can apply, there are a lot of web tools available. You can place your sequence there. It automatically identifies the best target sequences. So once you identify the target sequence in your genome, then next step is to generate the constructs. So as I mentioned before, we have this left tDNA border, right tDNA border. In between, we have to incorporate our reagents. So one thing, important one is this selectable marker. Whatever the cell has taken up this tDNA, we have to be able to identify that. In this case, we utilize this hygromycin. We have the Cas9 and we have sgRNA acid. All these components we incorporated into the vector and mobilized into agrobacteria. Then we performed the plant transformation on these cotton tissues. After 26 days after infection, we can see still a lot of cells have this GFP expression. By 30 days, we are seeing that some of the cells are losing the GFP expression. So this is under the fluorescent microscope. Under this fluorescence, cells that have the GFP expression, they will be in green. Cells that lost the expression because of the chlorophyll, they are emitting this red color. So all the red cells have lost the GFP expression. So we continuously saw this progression, and this is at 70 day, we excise those tissues and continued to grow them. And you can see this under the bright field, you can see how the tissue looks and under the fluorescence. So this one is the completely silenced calli. This no silencing occurred at here, but here we can see this chimeric, half of the calli lost the GFP expression, only half of that has that expression. So later at genetic level, what are the changes we wanted to see? We applied the at 120 days after transformation, T0 plants were developed, tiny T0 plants. From that, we isolated the DNA, amplified this region from here to forward primer, reverse primer. We amplified this region and sent for Sanger sequencing. And these are the Sanger sequencing results. So these kind of mutations we can see, identified whatever changes that are happening in that plant. So this work is published in 2017. This is the first article that demonstrates the CRISPR-Cas9 technology in cotton. So we also did similar work in potato. So here you can see that these are the tissues. This is again a transgenic plant, which is having the same GFP. We utilized same constructs and applied this, and we found similar results even in potato. This technology is working. So next step is to identify whether this CGF genes, all three genes, we will be able to knock out and see the phenotype or not. So this is the CGF1 gene, which is located in the D chromosome, 11th chromosome. CGF2 gene is located in the chromosome 1. CGF3 is located on the chromosome 12. We selected these are the targeted sequences, and we generated these constructs like this. And these are the, after generating the transgenic plants, these are the phenotypes here. Compared to control plant, you can see this construct, 236 construct and 237, you can see either reduced number or completely silenced ones. So whatever you see these tiny dots here, this is still from the T0 plants. When we took into the T1 generation, we lost completely all these glands are missing from those plants. So we did the HPLC analysis. All these terpenoids are almost near to zero. And this is the actual phenotype in the seed. These are the control seeds, CGF2 silenced, and this is the CGF3 silenced seeds. So that's how we established the CRISPR-Cas9 technology and identified these genes. So further we can improve. And right now we are applying these same genes and trying to develop insect-resistant plants. Utilizing this information we found previously, utilizing that we can improve more insect-resistant plants we can develop from these genes. So we got the patent, and if anyone is interested, this is a nice article you can find in the Plant Biotechnology Journal. So that's all about my work from the cotton. That's the example from the gene discovery to all the way into the genome editing and how we proved that those three genes are involved in the gland formation. So right now, in particularly to the horticultural crops, this CRISPR-Cas9 technology has been applied to many of the species already. And of course, some of the vegetables are especially that we grow in Andhra Pradesh or India, all the protocols may not be available. So to such things, we need to start with the plant tissue culture, DNA delivery methods, and plant developing the transgenic plant, and eventually test this CRISPR-Cas9 technology. So on this slide, as you can see, there are a lot of fruits and berries, ornamentals, all these, they already tested the CRISPR-Cas9 technology. And there are many traits that we can apply to improve the crops, in especially with the horticulture crops, either biotic stress, abiotic stress, and enhanced breeding time, food security, we can apply these, and enhancement of a quality yield of the crop plants, we can apply. This is a nice review article where they discuss all these different traits. So by learning from all these model crops and higher end crops, we can apply these technologies to the horticultural crops. So these are my acknowledgements. Throughout my journey, a lot of researchers helped to grow into this role and develop all these technologies. I sincerely thank all of them. And I thank all of you for attending this seminar or the guest lecture. And I thank VC and Dr. Padmalata Madam for giving me this chance to present here.
Thank you very much, sir. And you have given very excellent and insightful presentation on genome editing and their applications in horticulture, sir. Thank you very much. Now the topic is open for a discussion, and any queries are there from the participants, they can get them cleared by raising your queries.
Sir, first my question. So excellent presentation. I am congratulating you that excellent work done in cotton and potato. So here my question is, you are transferring the DNA of agrobacterium in cotton. So after that, you are using agrobacteria as a vehicle to transfer that DNA into the cell. Is it correct or not?
That's correct,
Okay.
sir. Yeah. Yeah.
So, like satellite, putting the satellite in space, aircraft
Exactly.
is used as a vehicle. So, like
Exactly.
that, agrobacteria is a vehicle to transfer that DNA into cell. So, in
Correct,
case of
sir.
horticulture crops, suppose one variety is there that is susceptible variety. For example, any viral diseases, any diseases, it is susceptible. To suppose in tomato, bacterial wilt is the major problem, I think. Bacterial wilt is the major problem. So to transfer that gene, how to do that through gene editing?
So some of the things are possible with genome editing, and some of the things that we cannot apply genome editing. So I will tell the differences. Like I gave one example of the BT cotton. So the gene is available in the bacteria, but we need to bring that gene into the plants. So we have taken that gene and incorporated into the tDNA, and agrobacteria is able to deliver that into the plant cells.
Yeah.
So to improve in this example, if we want to develop resistant varieties, if we already know the gene, if we know that if we knock out that particular gene, because there are some genes that are susceptible genes will be there. There are some genes that are resistant genes. If we know the susceptible gene and if we knock out those genes, we can eliminate that. But the first step for that, the exact gene information has to be there. For example, tomato is very well known. We have the genome sequence available. Most of the genetic genes, all the information is available, so we can easily target. And some of the crops like bendi or brinjal or some rare crops, we cannot apply until we know the information. We cannot apply genome editing.
So that means the genome sequencing we should know that all crops. Suppose that is a viral susceptible gene, some varieties resistant genes will be there.
Yeah.
So by gene editing, we can replace that susceptible gene. You see,
Yes.
it it is a susceptible, is having good quality, high yielder, remaining all characters are good. But the
Yes.
thing is the susceptible nature. That
Yeah.
susceptible gene we can identify and can be replaced with resistant gene, taking from
Yeah.
other source.
Yes,
Taking
we
from
can
other source.
replace that. If we replace from other source, for example, we are bringing from other organism, that will become the transgenic plant. That
is
we are overexpressing the genes there. That will become transgenic technology, and for that we need a lot of deregulation processes. For
Okay.
genome editing, only few nucleotides we can change. Genome editing is equivalent to the mutant breeding. Like in
Mutant.
1920s, they started all the mutant breeding. So if we apply those irradiation or other technologies, it is all random. We don't know. We have to screen a lot of germplasm. But with genome editing technology, we know what we are targeting. We know where we are making that change, so we can precisely, lot of time we can save, lot of resources we can save, easily we can generate the desired trait once we know that information.
So what is the suppose for establishment of lab that gene editing? Because in India, so there is almost 140 crores population is there throughout the world. This agrarian agriculture dominant country and states. So this is the important technology. So the faster CRISPR is the faster technology, speed technology. So within 1 year we can get the transgenic plants and transgenic varieties or crops. So, so the approximate cost of this lab establishment and to reach the final stage, final transgenic plant, what is the appropriate that cost of establishing lab and other equipment?
Yeah, it
Roughly,
all depends on what is the scale that we are planning here. So
yeah,
for a
here,
simple laboratory, any biotechnology lab that we already have, in that we can start the laminar hoods and the growth chambers, walk-in growth chambers, these kind of basic equipment we can have. And the students or the faculty need to get trained first. Having the equipment will not help us to develop these
yeah,
plants. They need to go to the right place and get the right training. For
yeah.
at least 2 to 4 years, they need to get that basic training. Once they have the training and having the foreign collaborations who already have this technology will help to guide them, and easily they can success in their future
Yeah,
goals.
markers and all of that biotechnology, when in common state, that at vegetable research station, we have developed this markers and all biotechnological work with all equipments. After state bifurcation, we lost it. So, so establishment of lab is one aspect. Training
Yes.
at international level, that is to
Yeah.
faculty and students, is another aspect. So generally, okay, we can send our faculty to foreign countries like USA. There they can get the training of 1 or 2 years. They can hear and they can do the work. So we have already tissue culture labs and producing banana tissue culture, everything we have. That biotechnology lab, especially molecular biology labs, we have to establish. I think we are spending crores of rupees for different purposes. So I think for the establishment of lab, can you help in establishment of lab and gene editing and all transgenic plants production of vegetables and fruits? Because fruits is a very perennial nature. So we need a canopy that much size for high density planting. Instead of going for pruning manually, that if we can develop a bushy type of mango, we can go for many plants and high density planting. And if high yielder, quality, storage, the storage life can be increased. So you see the quality, we can get more yields, more quality products. So this is the era of international globalization for export purpose also. The resistant varieties. So because every health conscious that chemical residues more that's why many our shipment are rejected by our chilies or grapes. So many
Yeah.
products are
Yeah.
rejected. So high residues. So this is the one of the important area. If we can develop the resistant varieties for the biotic and abiotic and reduce the load of this chemical sprayings. So this is one of the important area. So definitely your services, your expertise, your knowledge will be utilized by YSR Horticulture University in future. And if possibility is there, we can have one MOU with our university. So you discuss with your people, and really excellent presentation you have done in cotton and potato. So like that, we want to establish in horticulture university that gene, because it's faster through conventional methods is taking a lot of time, especially in perennial crops and vegetables. So that's why this is the very fast CRISPR. And any biotechnological labs in India is already established. Have you have any idea of in Indian context?
ICR institutes, they already have, to some extent, and especially in Delhi, many of the labs already have this technology.
Yeah.
Yes, sir, they already have it. Yeah.
They already having. Okay.
Yeah, they are working on it. Yeah. But
Mm.
not on horticulture crops, like majorly, like I believe that I reviewed several articles as well. Rice they did already, tomato and major crops they already started working on major crops. Yeah, some of the crops like cotton, these are difficult ones. Horticulture crops are also difficult ones. We don't have that technology yet into those crops in India. But yeah.
You are telling that horticulture crops are not are difficult. What are the reasons between cotton, for example? So biology, all plants are the same. That growing, responding to environment and all is same. Why the horticulture crops are difficult in gene editing? What are the reasons?
Gene editing wise, once we have the reagents in the cell, the genome editing will occur. But how we are going to send into those cells? So, for example, like you mentioned, how we are sending the satellite into the space. Are
Yeah.
we going to reach to the moon yet? Are we going to reach the Mars yet? That is the difference between the rice and any other horticulture crop, because agrobacteria has to be able to interact with that particular species. For example, in cotton, in my one of the slide, I showed that different crops and what are the genotypes. If I have to work on other genotypes within the same cotton cocoa, the transformation process is working well. If I take other genotype or Indian genotype, I cannot do transformation that easily. So that challenge is still there. But right now we are applying the second generation regeneration techniques where that is not having any limitations. As long as agrobacteria is able to deliver regeneration wise, we are good. So there are two stages we need to consider. Plant transformation is one, and how we are going to deliver into the cells is another aspect. Plant transformation we can achieve maybe we applying these developmental regulatory genes, but delivering DNA also is another challenge. So once we figure out those two challenges in any crop, we can establish this.
Yeah. So
It all requires a lot of modifications and exploration and identify the precise method where we can achieve higher efficiencies.
Yeah, if this technology is successful, because all horticulture crops are very perishable in nature, that
Yes.
we cannot keep more many days. So definitely this will help horticulture crops. Anyhow, thank you very much for hearing today, and we got so much information on gene editing. Okay, invite any other members, university officers, and all. You can ask the questions and clarify it.
Any questions from the participants?
Sir, good morning, sir. Good morning, sir. Nice presentation, sir. Sir, you explained crown gall infestation in grapes, sir. Here in Andhra Pradesh and Telangana states, this crown gall disease is more severe, sir, in flower crop, that is rose crop, sir. This agrobacterium mediated transformation mechanism is similar In case of rose also, sir, is there any commercially available agrobacterium Rhizobacter in US? Is there any commercially available, sir? Here there is no commercially available products here for controlling this bacterial diseases, sir. And one more thing, this you explained antibiotics, kanamycin and hygromycin and herbicides. Here we are using kasugamycin along with copper oxychloride, sir. This kanamycin it is also for controlling this bacterial diseases, sir.
No, madam. Antibiotics that I explained is to select the tissues that have taken up the tDNA. This is not to
sir.
control any, because these antibiotics are very expensive. We cannot
Yes, sir.
apply at field level to control the
Mm.
bacterial diseases.
Oh,
This is
okay,
only
sir.
to select the plant tissues
Mm.
that have taken up the
Mm.
tDNA. And to your
Mm.
first question about the crown gall disease, the
sir.
management strategies are the only choice that you can apply. Unless you develop any transgenic plants, it's not,
Sir.
we cannot avoid that. You
Okay,
cannot
sir.
avoid the
Here
crown gall
first
disease.
removal and destruction of those infected plants, sir. That is only
Exactly.
mechanism. And
Only
also,
mechanism right now.
mm,
Yeah. Otherwise, you
right.
need to develop the transgenic plant where it can withhold the this one,
Yes,
or
sir.
even you can apply the CRISPR technology as long as
Sir.
the
Yes, thank you.
you can apply the CRISPR-Cas9 technology as well to remove those crown gall related
Infected.
genes. Yeah.
Thank you, sir. Thank you, sir.
Madam,
Thank you,
good
madam.
morning, madam. With regards to your question on Agrobacterium tumefaciens, University of
Sir.
Horticulture and Forestry, Himachal Pradesh, they have developed Agrobacterium radiobacter strain. So they have
Yes.
commercialized it. If you want to test means, we can write to that particular Solon University, and we can try against crown gall of rose also, madam. Even when I was working at Madanapalli, I have seen some crown gall diseases in rose and other crops. Like because we have very limited options for controlling the bacterial diseases in horticulture crops, like agrobacteria, we can try its efficacy in our region, madam.
Yes, yes. The Agrobacterium radiobacter is available in the third year course also explained to the students. That's why I am asking.
This is Indian
Okay.
strain, madam. That Agrobacterium
India.
radiobacter UHF, University of Horticulture BA 218. So we can write to that university and we can test against our diseases here, madam. Agrobacterium like crown gall of rose or anything. That's it.
Okay. Okay, sir. Thank you, sir.
Thank you.
Sir, other than agrobacterium for transformation of gene, are there any other bacteria or any other organisms available, sir? Only you are using only agrobacterium, and why it is used?
So for agrobacteria, we know the mechanism very well, how to utilize that tDNA mechanism. That's why people are very much interested to utilize this particular agrobacteria strain. In 2022, they published another article where they are using a different, it's called Archobacterium. So they are using that bacterium, but we still don't know the mechanism very well. And availability of the strain is also important. Lot of patents will be there. Agrobacteria, we can apply. Patent has recently expired, but others, it's not easy to get the strain or apply. And in case of mechanical methods like gene gun is the choice. In animal sciences, they apply the PEG mediated, chemical mediated, all these different types of transformations. But in plant cells, because we have the thick plants cell wall, it's not easy to transfer. We need either mechanical force or biological interaction is needed.
Okay, sir. Thank you.
Sir, this is Jayaprada. Yes, sir. Sir, you mentioned that CGF3 gene, which silenced CRISPR-Cas, that black glands, no, sir?
Mm.
In the same way, and you also mentioned the speed of germination from wheat, the gene can
Yeah.
modify, speed up the germination in cotton. Can we apply the same to
Yes, yeah, we can
artificial?
apply, we can apply those similar technologies like in maize. They applied the baby boom mussel. So it is a patented technology right now, but for research purposes, we can still apply those genes. They they will not work equally as what they tested in maize. If you utilize the baby boom mussel in the dicots, the efficiencies will not be that greater. So a lot of parameters has to be tested and optimized. But compared to the traditional applying the hormones externally, these DR genes will definitely help.
Okay. Thank you, sir.
Yeah.
Any more queries?
Good morning, sir. This is Dr. G.
Good
Lakshmi
morning.
Devi, Assistant Professor in Biotechnology, sir. Sir, excellent presentation, sir. I need small information regarding the target gene sequencing identification because banana and tomato are gene sequencing already available, sir. Even agrobacterium mediated transformation also available. I'm also having the experience in transformation, implanted transformation in IIOR. But only problem here is to identify the target sequence. If I want to develop any abiotic stress or something, any physiological changes, I need to take the target sequence identification, sir. If you enlighten me regarding these softwares, it will be helpful, sir.
Yeah. So basically, you can approach this into several different ways. You have to collaborate with the breeders. If you already know the near isogenic lines, that will be the best approach to perform the whole genome sequencing, RNA sequencing analysis, and identify the mutations in certain genes and test out those genes. The second approach is from the literature. You have similar disease in other crops or any such information is available, you can utilize that information and target those genes.
Okay, sir. Thank you, sir. Sir, any like softwares available means you are telling like already known information only you can take the and catch it with the reference genome like that.
Software wise, you need to apply the NGS technologies like RNA-seq analysis. There is a whole pipeline to perform that. Like if you read my article in the Plant Biotechnology Journal, you will see the what are all the different softwares I applied. But basic thing, you need those two near isogenic lines. Once you have those, you need to isolate the RNA from those tissues and send for the sequencing. Once you get the sequences back, you need to have the bioinformatic tools and you need to have that particular server established. All those tools you need to be able to install, and you need to have that Linux all on the Linux environment. You need to install all those software. Basically, any person who is doing the basic bioinformatics, they can help you to to identify the differential expressed genes.
Okay, sir. Thank you, sir. I will go through your paper and I will.
Yeah.
Thank you.
Sir,
Feel
one
free,
more
if
thing.
you have any questions, you can always ask me.
Yeah, one more thing I want to ask
Yes,
that how to integrate artificial intelligence with the genome editing.
So right
we-
now, my perspective I'll give. Right now, artificial intelligence applying to genome editing technology, we are really far away, I feel, because how are we going to identify the genes? That is the first step. So with the current AI, even I am interested to apply the machine learning methods. Recently, we wrote one review article on that, and one of the research article also we are developing right now, applying the machine learning methods. So by applying machine learning methods, we need to have large data, so many samples, RNA-seq data we need to have, identify those. It's very complicated. We are still in that very early baby steps, like developing all these and training the models. All that we are still need to learn how to achieve that.
Okay, if there are no more queries, now I request Registrar Madam to propose vote of thanks. Madam, Registrar.
Thank you, Madam. Respected Dr. K. Dhananjay Rao, Honorable Vice Chancellor, distinguished guest speaker, Dr. Madhusudan Reddy Janga, esteemed university officers, eminent heads of institutes, faculty, and students, a very good morning. It is my privilege and pleasure to propose vote of thanks on the occasion of guest lecture on plant genome editing and its applications in horticulture crops improvement under 31st Global Connect series. At the outset, I express my sincere gratitude and thanks for providing great support and encouragement for organizing such academically enriching programs to our Honorable Vice Chancellor, sir. On behalf of University, I express my sincere gratitude to our esteemed guest speaker, Dr. Madhusudan Reddy Janga, Texas Tech University, Lubbock, Texas, USA, for taking time to be with us and for sharing valuable insights into the rapidly advancing field of plant genome editing. The lecture has provided us with an excellent understanding of modern genome editing technologies and their potential applications in developing improved crops with desirable traits. Thank you, sir, for the enlightening and informative lecture. I also express my gratitude to deans, directors, ADs, ADRs, principals, scientists, faculty, and students for their enthusiastic participation, meaningful interaction, which made today's program highly successful. My special appreciations to the organizing committee, Dr. T. Padmalata, Director of Industrial and International Programs, technical and administrative staff, whose efforts contributed to the successful conduct of this lecture program. I am confident that the knowledge and perspectives gained from today's lecture will inspire our faculty and students to explore new avenues of research and innovation in crop improvement. I also thank everyone present over here for their valuable participation and for making the program a meaningful and successful one. Thank you all.
Thank you, Madam. So on behalf of Dr. YSRHU, I thank Honorable Vice Chancellor, sir, Dr. Madhusudan Reddy Janga garu, and all other faculty members for joining this Global Connect webinar. With this, we conclude today's session.